Did you know that we have an entire ecosystem of microorganisms living in our gut? While these microbiota are essential to life, an imbalanced state can lead to the development of GI cancers. It's important for radiologists to be familiar with these as we play an important role in the diagnosis and management of these cancers. Listen on to hear a short summary of the July-August GI radiographics review by Dr. Bondi et al. On human gut microbiota-associated gastrointestinal malignancies. For the whole scoop, make sure to check out the full article at the link. The human gut contains anywhere between 500 to 1,000 different species of microbes. Most are harmless and actually benefit us by two major functions. The first is by immunomodulation and helping to establish a mucosal immune system. The second is by nutrient production, such as B vitamins and vitamin K, as well as assisting in degradation of various compounds. Normally, we have a balance in these healthy microbes, but if an imbalance in the gut microbiome occurs with a decrease in our normal gut flora and an increase in abnormal microbes, this can predispose to the development of various cancers. There are three speculated theories for the pathogenesis of these microbiota-associated cancers. The first is activation of the tumorigenic pathways and suppression of tumor suppression pathways. The second is a pro-inflammatory signaling resulting in chronic inflammation of the epithelium. And the final pathway is direct cytotoxic effects resulting in damage to the host DNA. All of these result in abnormal GI epithelium and can eventually lead to these gastrointestinal malignancies. So let's start by talking about a couple of specifics. We'll start with the esophagus. 60% of esophageal cancers in the US are adenocarcinoma. In the setting of chronic gastroesophageal reflux disease, the natural esophageal flora, primarily composed of gram-positive bacteria, become unbalanced and become mostly gram-negative bacteria. These new bacteria release a lipopolysaccharide endotoxin, which activates pro-inflammatory and tumorigenic pathways and eventually can result in adenocarcinoma by activating cytokines such as interleukin 8 and TNF alpha. As many patients will present with symptoms of dysphagia, heartburn, or vomiting, these cancers may initially be detected by radiologists on fluoroscopic barium swallows, as shown here. Endoscopic ultrasound is the modality of choice for assessing the depth of penetration, also known as the T stage, while contrast-enhanced CT or FDG PET CT are used to complete staging and for follow-up. Interestingly, there's some evidence that prebiotics and probiotics can actually help prevent esophageal adenocarcinoma and can improve the effect of chemotherapy, respectively. Moving on to the stomach. Of the two histologic subtypes of gastric adenocarcinoma, the intestinal subtype is associated with infectious agents, including H. pylori and Epstein-Barr virus. H. pylori produces a protein called CAG-A, which degrades the p53 tumor suppressor gene and leads to the activation of multiple chemical mediators, including cytokines. These lead to a sequence of changes starting with gastric atrophy, chronic inflammation, then metaplasia, then dysplasia, and finally adenocarcinoma. Figure 4 from the article shows a polypoidal mass protruding into the lumen of the stomach seen on a fluoroscopic upper GI series performed for epigastric pain. Figures 9 and 10 show contrast-enhanced CT of two different patients showing an intestinal gastric adenocarcinoma. Interestingly, again, probiotics have been shown to help in preventing precancerous states with H. pylori-associated gastric cancer. Moving on to another microbiota-associated gastric malignancy, and H. pylori strikes again. H. pylori is not only associated with the development of gastric adenocarcinoma, but is also a causative agent in the majority of gastric mucosa-associated lymphoid tissue lymphomas, also known as MALT lymphoma. MALT lymphoma accounts for most of the gastric lymphomas, and it's important to differentiate them from other lymphomas as the clinical course and management are entirely different. And if diagnosis is delayed, high-grade transformation can occur, which results in a worse prognosis. Eradication of H. pylori is actually the mainstay of treatment, although some cases may require additional chemotherapy, radiation, or immunotherapy. This is an example from the article on Figure 13 showing a malt lymphoma on contrast-enhanced MRI and endoscopy. Probiotics have been shown to be helpful in the eradication of H. pylori and to halt progression of malt lymphoma in animal models. Moving on to colon. While mortality due to colorectal adenocarcinoma has actually declined in the past decades, there has been a sharp increase in incidence in patients younger than 50 years old, with some etiologies including possible early life carcinogenic exposures, including gut dysbiosis. All of these microbes have been implicated in the development of colorectal adenocarcinoma. For example, S. bovis is thought to cause a release of various cytokines, including COX-2, IL-8, and NF-kappa beta, and it can result in the inhibition of apoptosis and stimulation of angiogenesis. Similar to other malignancies, probiotics may reduce chronic inflammation and tumorigenesis that is implicated in the colonic microbiome dysbiosis and can even serve as an adjunct during anti-cancer treatment, potentially reducing severity of GI side effects from chemotherapy. And last but not least, and perhaps one of the best known microbiota-associated malignancies is anal squamous cell carcinoma. More than 90% of anal squamous cell carcinomas are associated with HPV infections, especially HPV 16 and 17. E proteins produced by virus cells suppress cell cycle regulatory tumor suppressor proteins P53 and RB, which eventually leads to the transformation of anal epithelial cells into anal intraepithelial neoplasia and eventually anal squamous cell carcinoma. And this is where the HPV vaccine comes in, and it has shown reduced rates of AIN and squamous cell carcinoma. So what can we do to ensure a happy gut flora? Well, probiotics, such as those belonging to the genus Lactobacillus, may offset the negative effects of these unpleasant microbes by indirectly reinforcing the mucosal gut epithelium and reinforcing the seal between epithelial cells. They also produce short-chain fatty acids that downregulate the inflammatory cascade that can result in carcinogenesis. Finally, fecal microbiota transplantation, also known as FMT, is an emerging technique to restore the protective gut microbial barrier. I hope you enjoyed this short info video all about the microbes that live in our gut and how gut dysbiosis can lead to various GI malignancies. Make sure to check out the full article at the link.